Chapter 11 Photosynthesis in Higher Plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have already learned that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, a physico-chemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is the primary source of all food on Earth. It is also responsible for the release of oxygen into atmosphere by green plants. Have you ever thought what would happen if there is no oxygen to breathe? This chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into chemical energy. .1 What do we know? Let us try to find out what we have already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, light, and carbon dioxide are required for photosynthesis to occur. You may have carried out experiments to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in green parts of the leaves in the presence of light. Another experiment you may have carried out where a part of leaf is enclosed in a test tube containing some potassium hydroxide soaked cotton which absorbs carbon dioxide while the other half is exposed to air. The setup is then placed in light for some time. On the testing for the presence of starch later in the two parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch, while the portion that was in the tube tested negative. This showed that carbon dioxide was required for photosynthesis. Can you explain how this conclusion could be drawn? 11.2 Early Experiments. It is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: Plants restore to the air whatever breathing animals and burning candles remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, Jan Ingenhousz showed that sunlight is essential to the plant process that somehow purifies the air fouled by burning candles or breathing animals. Ingenhousz, in an elegant experiment with an aquatic plant, showed that in bright sunlight, small bubbles were formed around the green parts, while in dark they did not. Later he identified these bubbles to be of oxygen. Hence, he showed that it is only the green parts of the plants that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. His later studies showed that the green substance, chlorophyll, as we know it now, is located in special bodies, later called chloroplasts, within plant cells. He found that the green parts in plants is where glucose is made and that the glucose is usually stored as starch. Now, consider the interesting experiment done by T.W. Engelmann. Using a prism, he split the light into its spectral components and then illuminated a green alga, Cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of the photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B. By the middle of the 19th century, the key features of plant photosynthesis were known, namely, that plants could use light energy to make carbohydrates from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood as carbon dioxide plus water in the presence of light gives carbohydrate, the general formula CH2O plus oxygen, where CH2 represented a carbohydrate, that is glucose, a 6-carbon sugar. To the understanding of photosynthesis was that made by the microbiologist Cornelius von Mehl, who, based on his studies of purple and green bacteria, demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by 2s2a plus carbon dioxide, that is CO2, in the presence of light gives 2a plus CH2O, that is carbohydrate, and water. In green plants, S2O is the hydrogen donor and is oxidized to oxygen. Some organisms do not release oxygen during photosynthesis. When hydrogen sulfur, when H2S instead is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur or sulfate depending on the organism and not oxygen. Hence, he inferred that the oxygen evolved by the green plant comes from water, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6 carbon dioxide plus 12 water molecules in the presence of light give rise to glucose, that is C6H12O6 plus 6 water and 6 molecules of oxygen, where C6H12O6 represents glucose. The oxygen released is from water. This was proved using radioisotope techniques. Note that this is not a single reaction, but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrates are used in equation given above? .3 Where does photosynthesis take place? You would of course answer in the green leaf or in the chloroplasts. Based on what you earlier read in chapter 8, you are definitely right. Photosynthesis does take place in the green leaves of plants, but so also in other green parts of the plants. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually, the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplasts will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is a membranous system consisting of grana, the stroma lamellae, and the matrix stroma. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn forms starch. The former set of reactions, since they are directly light driven, are called light reactions or photochemical reactions. The latter are not directly light driven but are dependent on the products of light reactions, that they are ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reactions, carbon reactions. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. 11.4 How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves, even in the same plant? We can look for an answer by trying to separate the leaf pigments of any green plant through paper chromatography. A chromatographic separation of the leaf pigments shows that the color that we see in leaf is not due to a single pigment but due to 4 pigments. Chlorophyll A, which gives bright or blue-green in chromatogram. Chlorophyll B, yellow-green. Xanthophylls, yellow, and carotenoids, yellow to yellow-orange. Let us now see What roles various pigments play in photosynthesis? Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A pigment to absorb lights of different wavelengths according to figure 11.3a. Of course, you are familiar with the wavelengths of the visible spectrum of light as well as the Vibgyor. From figure 11.3a, can you determine the wavelength or color of light at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelengths too? If yes, which one? Now look at figure 11.3 showing the wavelengths at which maximum photosynthesis occurs in a plant. Can you see that the wavelengths at which there is maximum absorption of chlorophyll A, that is blue and red regions, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 11.3c, can you say that there is a complete 1-to-1 overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together show that most of the photosynthesis takes place in the blue and red regions of the spectrum. Some photosynthesis does take place at the other wavelengths of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophylls, and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelengths of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation.